Our next speaker is Dr. Randy Nelson. He's a professor and chair of the Department of Neuroscience at Ohio State University. He holds a John and Olive Broomberg Chair in Brain Research and Teaching and directs the OSU Neuroscience Research Institute. He has over 400 publications on the topic of biological rhythms, behavioral neuroendocrinology, stress, immune function, and aggressive behavior. Dr. Nelson.
Thank you. And thanks for inviting me to this. This has really been a great experience. Thank you. So I'm going to go really fast because I have a lot of stuff because I'm covering mouse models of not cancer, which covers a lot. And I need to learn how to make this work. No, it doesn't work? Okay, I'll just use this. Okay, we've all seen that the problem with modern circadian disruption is profound, and we've just come to appreciate it in the last few years. But the Catholic Church, which has always been progressive, actually banned streetlights in 1831, noting that God very clearly established the delineation between night and day, and you shouldn't mess with that. So kudos to the Church. But certainly there's been 3 to 4 billion years of evolution of life with dark nights and bright days. In the past 130 years, we've messed with that. And so about 10 years ago or so, Kristen Navarro and I published this, what might be a cause of concern of what light at night might do. And so I'm going to cover just 4 things: energy metabolism, add one mood that Richard just mentioned, cardiovascular events, and immunity. Okay, so I'll cover those 4. We're going to use the same kind of model in each one of them. So we see the effects in humans of people on night shifts eat more, and the metabolism tends to be jacked. And we see an increase of light pollution and obesity in this country and elsewhere in the world. So the model basically is light at night disrupts circadian rhythm, however you wish to define that, causes brain changes, and then affects body mass regulation. We measure lots of different things associated with that. In this model, I'm going to talk about with these 4 different areas. It's all going to be the same. We're going to expose the animals to very dim light, 5 lux of light at night, white light, and I'll show you some exceptions to that. And that falls somewhere between full moonlight and twilight. Okay, so if you put animals, these are mice, in dim light at night, they increase their body mass by about 15%. And whether they're in dim light at night or constant light, they gain body mass relative to animals in dark just over the course of 8 weeks. It doesn't take long for this to happen. And over the course of 8 weeks, they start to show a pre-diabetic state in the sense that glucose processing, glucose tolerance test is impaired. It's a pre-diabetic thing. And if they're given a high-fat diet, they also show increased insulin too. Again, this is just after 8 weeks. It's not a stress response. We don't see a change in glucocorticoid between dark night or dim nights. If they're in constant light, of course, the circadian rhythm gets masked a bit, and you sort of have moderate glucocorticoids throughout the day. But this body mass increase occurs when there's no difference in locomotor activity, so they're exercising the same, and they're eating the same number of calories, which seems to be breaking some rules. And as Dr. Turek mentioned yesterday, what they're doing, however, is they're I won't say voluntarily, but in response to the dim light at night, they're changing the time of their food intake. So they're eating more of their food at the wrong time of day. So to summarize that, light at night makes you fat. Okay. So as Dr. Turek mentioned, it seems to be the timing of the food intake that differs. And if you look at the animals in constant light, so constant bright light, like maybe some of our shift workers, you can see they're eating about 80% of their calories at the wrong time. And so what we did, as Fred mentioned yesterday, we limited their food access. And so if you put animals that were in dark nights but you gave them food at the wrong time, then they gained body mass, as they had reported a couple years before. And if you have animals in dim light, but you only let them eat during the correct time, then they don't gain the weight. So that might be something to think about in terms of, Dr. Pandas had a very nice study on the mechanism of this, just published recently in Cell Metabolism. Basically, by restricting the time of when the animals ate, even though they had a high-fat diet, they didn't develop all the bad things associated with a high-fat diet. So that was great. And I didn't need to show this because Fred showed it yesterday, but this was sort of the first vision of timing of food intake drove all these things. And what we're arguing is it's the dim light at night that causes the animals to eat the wrong time. Okay? Now, where I diverge from the common opinion around here is that I don't think it's the sleep. People say, do you know that mice are nocturnal and humans are not? And I do. So if reviewer two's out here, I do. But we chose these guys on purpose. We have diurnal species in our lab because they sleep during the day. And so we assumed that they were not being affected in their sleep. And so we had to run this paper, and thanks Dr. Smolensky for publishing 8 graphs of null results. But dim light at night does not affect sleep. So I think what happens is the main biological clock gets affected, sleep being one of the hands of the clock that gets affected downstream in humans perhaps, melatonin another hand of the clock. These mice also don't have melatonin, so it's not a melatonin-mediated response. And again, we did that on purpose. There are strains of mice that have melatonin, not this one. Okay? So what's happening? What's happening is the actual mechanisms of the clock themselves, the clock genes, are being affected. So here's this protein expression. Here's period 1, and you can see the amplitude of the peak is suppressed in dim light at night. Okay, similar, you see a sort of a shifting of the peak, but basically amplitude is suppressed. And so one target might be these small molecule and circadian modulators that people like Jake Chen are working on in Takahashi that you might be able to give that would increase the amplitude of these substances. Clock and BMAL, my pet hypothesis was clock, what we were doing with dim light at night was suppressing clock, basically getting Fred's knockout, and that was causing the obese phenotype. But clock's not affected in this model. And we can look out on the periphery, and as Dr. Turek has talked about, out here in the white adipose tissue or in the liver, you see a lot of interesting effects. So you have to look at the peripheral clocks in these models too, and seeing that specific genes, again, period 1, period 2, BMAL being the major players as well as the cryptochrones. Okay, so that's energy metabolism. Let me talk about mood because Dr. Stevens brought it up as well. I think this is an important area. There we go. So the nurses' studies showed that night shift workers have more incidence of depression than people doing the same job during the day. The depression rates have increased in this country up to about 20%. In developed countries, 20% increase of 20% rate of depression. Among my Amish friends, they have the same rate of depression as we did in the 1950s, 1%. And that number has stayed stable. Just bring that up. Same kind of model again, 5 lux of light at night, looking at depressed mood. This series of studies done on Siberian hamsters for a variety of reasons are melatonin proficient. Again, they're nocturnal, so we didn't think we'd be affecting the sleep. And we worked on females because depression's reported 2 to 3 times more commonly in females than males. And we know there's this nice relationship, I don't know if nice is the right word, reciprocal relationship between depressed mood and changes in biological rhythms. So depression causes changes in sleep and melatonin and body temperature and those sorts of things. And also all the environmental perturbations that many of us have been going through can also cause depression. So you have this relationship. So how do you study depression in animals? Well, there's some tests that have been validated. One's called the Perceval forced swim test. The other one is Sucrose preference test. And so in the Perceval forced swim test, you put an animal in this bucket, and you see how fast they can swim, or how much they swim, how quickly they give up. And in terms of the anhedonia test, you see how much sugar of the water you take. So basically, how long they float, you can see in dim light they float more, they try to get out less, and they take in less of the sweet water. So that's a depressive model. We see the standard glucocorticoid pulses in the animals in dark nights, but that gets suppressed in, and the amplitude suppressed in dim light at night. So it just looks like that. We validated this. We looked at all the different markers of depression in humans. There's reduced spine density in the CA1s of dim light at night animals. Show me two things. And that correlates with the depressive behaviors. We see a reduction in BDNF in the hypothalamus of animals in dim light at night. We see increase in neuroinflammation, particularly tumor necrosis factor in animals in dim light at night. Yes, reviewer 2, it works the same in diurnal creatures. So we use these now grass rats, which are diurnal. You see exactly the same thing. Get off my back, reviewer 2. Okay. Cardiovascular. This paper just came out this week. Circadian misalignment increases cardiovascular disease risk, and we've been interested in light at night in vulnerable populations in hospitals and nursing homes. As we just heard, these folks don't get much light during the day, as well as getting light all night. And so here's light levels in intensive care unit in our patient rooms in our heart hospital. Our hypothesis was cardiac arrest, recovery, and outcome has to be negatively affected by this light at night. All right, and so we gave animals a cardiac arrest for 8 minutes, and what we see is they're in dark nights, and then for a week they're in dim light at night, 5 lux at night, and you can see the survival of those animals in dim light at night is remarkably impaired. And we just got a nice NIH grant from NINDS, thank you. If my PO is listening, he said he would be. Light at night increases hippocampal cell death significantly. So you have a cardiac arrest, you have an increase in cell death because of global ischemia. But if they had 7 nights of dim light at night afterwards, again, this is afterwards, you increase that cell death. You can see this in the floral jade. The jade color indicates dying cells. And all the markers are bad for inflammation if you have dim light at night. Okay, whether it's microglia or regular. And we just got these data literally 3 days ago. They're pretty cool, I think. This just shows active blood vessels. Active blood vessels and tomato lectin staining. And you can see that dim light at night reduces the number of blood vessels around the dentate, which I think is really interesting. All right, I'm going to go really fast here. She keeps giving me signals. Same thing happens for a focal ischemia. We have an hour. For focal ischemia, we do a middle cerebral artery occlusion, reduce survival, and increase in the infarct volume after a week of dim light at night. All right, so that's significant. How do we minimize the effects? We use red light. Dr. Brainerd's work showed that the melanopsin sensitivity is here in the short wavelengths, and so why not look at the red? So we compared white, blue light, and red lights, and basically red wavelength is much less detrimental in terms of the depression data, in terms of the neural response data, in terms of the cardiovascular data. So here's cell death. You see it goes up in dim light at night, comes back down. Here's survival, red light at night, the animals survive better. The neuroinflammation knockdown by dim red light at night. And it also works in the stroke damage as well. Okay, so these are safe lights in our lab. They're not really that safe, I would argue. And finally, let me just show you this quick data, because this is also very new and very exciting and very terrifying, I think, in some ways. So we know that dim light at night impairs a number of immune responses. This is delayed type hypersensitivity. These are in males. But you can see here's animals in dark, and here's animals in dim light at night. This is antibody production. It's increased. so not always impaired. But here's the cool study, or scary study, depending how you like it. We had male and female hamsters sitting in dark nights or dim lights, and then we paired them for a romantic encounter. And the lights went off, and they were exposed to dark nights the rest of the time. And the pups were gestated and born in dark nights, okay? And so some of the moms came from a dark night experience, some came from a light night and a dim light at night, and so on. So we wait for the offspring to grow up. They only have dark nights. Male offspring cell-mediated responses are impaired, dampened by maternal exposure to dim light at night, okay, which is not good. And paternal exposure to dim light at night dampens the female offspring to cell-mediated responses. And we don't know how this works yet. Again, these are very new data, but it looks like there's something going on with the glucocorticoid expression. There's a sex difference in what gets affected. So I think that's really interesting data, and maybe scary if our exposure to dim light at night, or because of shift work or whatever, is being imposed on our offspring. So take-home message. Dim lights at night derange metabolism. Might want to think about that verb for our definition. Depressed affective responses are evoked or provoked by light at night. Neuroinflammatory responses are increased by light at night. If you reverse the neuroinflammation, you effectively get rid of the increased body mass and depression responses. I don't want to talk about that, but you can change things with basically changing the light to long wavelength light in these animals, nocturnal or diurnal, and dim light at night exacerbates brain damage and impairs survival. And it looks like there's a transgenerational effect, at least in immune responses. Okay, so thank you very much for your attention. Here's the folks that did all the work and the funding agencies, which we're grateful to. Thank you for your attention.